All right, so get this.
Mm-hmm.
You folks have sent in a research paper on cryopreservation.
Yeah.
Specifically
Mm-hmm.
for stimulated T cells.
Right.
Pretty cool stuff, right?
Yeah, very cool.
It's basically like creating a pause button for these tiny but mighty immune cells.
Mm-hmm.
And our guide for this deep dive is a detailed report from Assay Genie,
Okay.
published just a couple weeks ago on November 6th.
Wow.
So
It's pretty recent.
very recent.
Yeah.
So
It's remarkable technology, really. I mean, think about it. Being able to preserve not just the cells themselves, but
Very-
functionality.
yeah.
That's like hitting save on a video game right in the middle of like an intense battle.
right. So let me ask you this.
Yeah.
What's the big deal about freezing these stimulated T cells in the first place?
Well, imagine you're a scientist trying to understand how the immune system fights off a virus.
Okay.
Stimulated T cells are like the special forces,
Okay.
primed and ready to attack. Cryopreservation allows researchers to study these cells later on their own terms
Yeah.
without having to go through that whole stimulation process again.
Yeah.
Talk about a time saver.
Right.
It also ensures consistency in their experiments.
But wouldn't freezing them cause some damage?
Right?
I mean,
Right.
I've lost my fair share of strawberries to freezer burn.
That's a great question,
Yeah.
and the answer is yes.
Okay.
Freezing can damage cells,
Hmm.
if not done carefully.
Right.
But researchers have developed specialized techniques to protect these delicate cells during the freezing process.
So how do they do it? Walk me
Big-
through this cellular time travel process.
Picture this. You're packing for a long journey.
Okay.
You need the right supplies
Right.
and a sturdy suitcase.
Okay.
Same goes for cells heading into the deep freeze.
Got it.
Before we even get to the freezing part,
Mm.
there's some crucial prep work involved.
Sounds intriguing. So how do we get these cells ready for their icy adventure?
First, we need to make sure the T cells are stimulated.
Okay.
Think of it like this.
Mhm.
They're sitting around chilling
Right.
when suddenly, bam, they're exposed to antigens.
Okay.
These are like the enemy signals
Got
that
it.
trigger the immune system to kick into gear,
Okay.
to boost things even further.
Mm-hmm.
We introduce cytokines.
Okay.
Imagine them as tiny text messages that cells use to communicate with each other,
Okay.
coordinating their attack against invaders.
Wow.
And sometimes we even use antibodies, which act like guided missiles,
Okay.
targeting specific cells.
Right.
This whole process ensures the T cells are activated
Mm-hmm.
and ready to go when they're thawed out later.
Wow. So it's a whole activation process before they even get close to the freezer.
Exactly.
But what about the suitcase you mentioned?
Ah, yes.
Yeah.
They need a special mixture called a cryopreservation medium.
Okay.
It's like a cellular antifreeze.
Right.
This medium prevents ice crystals from forming and wreaking havoc inside the cells.
Okay.
And it's not just about protection.
Mm-hmm.
It also provides nutrients
Okay.
to keep those cells happy and healthy.
So they're well fed and bundled up for the big chill.
Exactly.
That's impressive. What's next on the cryopreservation checklist?
Now it's time for the actual freezing process.
Yeah.
And this is where things get really precise.
Okay.
You need sterile cryovials.
Okay.
Think of them like tiny specialized test tubes
Okay.
and a way to control the freezing rate.
The article mentions a controlled rate freezer.
Yes.
Is that like a high-tech freezer
Yeah,
with
yeah.
all the bells and whistles?
Exactly.
Okay.
It allows researchers to slowly and carefully lower the temperature, preventing damage to the cells.
Okay.
But what's really cool
Yeah.
is that there's also a DIY option using a simple styrofoam box.
Hold on. A styrofoam box. That's incredible.
Yeah.
So even researchers without access to fancy equipment can do this.
Wow.
It's a testament to the ingenuity of scientists.
Yeah.
Finding ways to make groundbreaking research accessible to more people.
Right.
Pretty cool, huh?
Yeah, that's really cool.
Yeah.
So we've prepped our stimulated T cells,
Yep.
packed them in their special cryoprotectant suitcase,
Mm-hmm.
and we're ready to hit the freeze button,
Exactly.
whether it's with a fancy freezer
Right.
or the good old styrofoam box method.
Yep.
What happens next?
Well, now comes the delicate dance of freezing those cells. Whether you're using a high-tech controlled rate freezer or the ingenious styrofoam box method,
Right.
the key is precision.
Yeah.
It's like guiding a spaceship
Okay.
through a delicate maneuver.
Right.
You need to gradually lower the temperature to avoid any cellular mishaps.
So it's not just tossing them in the freezer
Right.
and hoping for the best.
Nope.
I guess those tiny cells are a bit more sensitive than my frozen peas.
Exactly. We're aiming for a slow and steady descent, to negative 80 degrees Celsius.
This gives the cells time to adjust to the decreasing temperature and prevents the formation of those harmful ice crystals we talked about earlier.
Right, right.
Remember, those crystals are like tiny daggers for cells.
So we've gently lowered the temperature to negative 80 degrees Celsius. Where do our cryonauts go next?
Now it's time for their long-term hibernation in the ultimate deep freeze, liquid nitrogen.
Wow.
We're talking negative 196 degrees Celsius.
Wow.
A temperature so cold that it practically stops time for these cells.
Wow. That's mind-bogglingly cold.
It is.
It's like a cryogenic spa for cells.
Yeah.
Where they can chill out indefinitely.
Yeah.
But speaking of chilling, I have to ask, what about thawing them out?
Right?
Is that as simple as popping them in the microwave?
Definitely not. Thawing is just as critical as freezing, and it requires a delicate touch.
Right.
We need to avoid those ice crystals forming again.
Right.
But we also need to be wary of something called osmotic shock.
Osmotic shock. That sounds intense.
Yeah.
What exactly is that?
Imagine a cell as a tiny water balloon.
Okay.
If you thought too slowly, water can rush into the cell faster than it can escape, causing it to swell and burst.
Oh, wow.
Think of it like a water balloon that's filled up too quickly.
Okay.
Can't handle the pressure.
Right.
And explodes.
Yikes. So thawing too slowly is like a cellular water balloon fight gone wrong.
Pretty much.
What's the right way to do it then?
Speed and precision are key here.
Okay.
The Assay Genie report recommends a quick thaw in a 37 degrees Celsius water bath.
Oh.
That's body temperature.
Okay.
So it's a gentle but rapid warm-up.
Okay.
You want to see just a tiny ice crystal remaining?
Okay.
That's your cue that it's thawed just right.
It's like a perfectly timed rescue mission.
Yeah.
Snatching them from the icy depths at just the right moment.
Exactly.
What happens next?
Once thawed, we need to ease these cells back into their normal environment.
Right.
We carefully transfer them to a tube containing pre-warmed growth medium.
Yeah.
Think of it as a nutrient-rich broth that cells love to live in.
Okay.
This helps to dilute the cryoprotectant and gently reintroduce the cells to a comfortable temperature.
It's like giving them a warm welcome back party
Yeah.
with a delicious meal.
Exactly.
But how long does it take for them to fully recover from their cryogenic slumber?
The report suggests giving them a good 24-hour rest period.
Oh, wow.
They've been through a lot.
Yeah, that sounds reasonable.
Yeah.
But let's face it, in the world of science, things don't always go according to plan.
Correct.
What could potentially go wrong during this thawing process?
Even with the most meticulous techniques, there are some potential pitfalls.
Okay.
Sometimes not all of the cells survive the freezing and thawing process.
Oh, no. So some of our little cryonauts might not make it back.
Yeah, it happens.
What causes that?
It could be due to several factors,
Okay.
like ice crystal damage, which highlights the importance of that slow and controlled freezing process we discussed earlier.
Right, right.
And even if the cells do survive, they might not function as well as they did before freezing.
So they might be a bit sluggish after their long nap.
Yeah, you could say that.
What causes that kind of post-thaw fatigue?
It could be due to something called DMSO toxicity.
Okay.
Remember that cryoprotectant we talked about?
Yeah.
While it's essential for protecting the cells, it can also be a bit harsh on them.
Okay.
Kind of like a strong medicine that has some side effects.
Okay.
Another possibility is that the cells didn't receive enough nutrients during the thawing and recovery process, leaving them a bit depleted.
So just like us, cells need a good meal and some TLC after a long journey.
Exactly.
Any other potential issues to watch out for?
The report mentions excessive clumping as another possible problem. This happens when too many cells are crammed together in the cryopreservation medium.
Right.
Imagine trying to pack too many clothes into a suitcase. Things get wrinkled and messy.
Okay, so we've covered the potential pitfalls.
Right.
Now I'm really curious about those expert tips and tricks you mentioned earlier.
Okay.
What can researchers do to ensure their cryopreservation endeavors go as smoothly as possible?
Good question. So you want the inside scoop?
Lay it on me.
Okay. The Assay Genie report emphasizes using fresh FBS and DMSO in that cryoprotectant cocktail.
Okay.
It's like baking a cake.
Right.
Using the freshest ingredients just makes a world of difference.
Right.
And remember, that 1 degree C per minute freezing rate we talked about, sticking to that gradual decrease is crucial for a smooth transition into the frozen world.
Fresh ingredients and a slow and steady cool down.
gut.
Yeah.
Yeah.
What about long-term storage?
Right.
Is that where the liquid nitrogen comes in?
Absolutely. Think of it as the ultimate VIP lounge for cells.
Okay.
It's the cryo equivalent of a luxury spa,
Right.
where those cells can hang out indefinitely,
Wow.
perfectly preserved at negative 196 degrees Celsius.
We talked about the importance of thawing quickly to avoid osmotic shock,
Right.
but I'm still a little fuzzy on what exactly happens there.
Yeah.
Can you break it down for me one more time?
Okay, imagine a cell as a delicate little water balloon.
Okay.
When you thaw it too slowly,
Right.
water molecules rush into the cell faster than they can leave.
Okay.
The cell swells up,
Yeah.
can't handle the pressure,
Right.
and it bursts.
Oh, okay.
That rapid thaw at 37 degrees Celsius prevents this watery demise.
So it's all about finding that sweet spot,
Yep.
not too fast,
Right.
not too slow,
Exactly.
just right.
So for
Protect those precious cells.
Yep.
What other tips do the experts recommend?
Patience is key.
Okay.
The report stresses giving those cells a good 24-hour rest period after thawing before putting them to work in experiments.
Okay.
They've just been on a wild ride
Yeah.
through time and temperature extremes.
Wow.
So a little R&R is essential.
It's like a post-vacation recovery period for cells.
Yeah, exactly.
Time to unpack, relax, and get back to their normal routine.
Exactly.
Another key point, minimize those freeze-thaw cycles as much as possible.
Yes. Very important.
Okay.
Each time you freeze and thaw those cells,
Right.
they lose a bit of their vitality.
Okay.
It's like making a copy of a copy.
Right.
The quality degrades with each iteration.
So handle with care.
Yes.
One freeze-thaw cycle per batch, if possible.
Exactly.
Are there any other tricks of the trade that stand out?
Yes. Adding a special ingredient called IL-2 to the growth medium after thawing
Right.
can give certain T cells an extra boost.
Gotcha.
It's like a little energy drink.
Right.
Helps them bounce back faster and perform at their best.
Little pick-me-up for those hardworking cells. And finally, the report highlights the importance of checking cell viability and functionality
Yes.
after they've had a chance to recover.
Very important.
It's like a post-flight inspection.
Yeah.
Making sure everything is in tip-top shape
Right.
before they embark on their next scientific mission.
Exactly. It's crucial to ensure those cells are ready for action
Okay.
before using them in important research.
So there you have it. We've gone from freezing those tiny cells to thawing them out. And we've even learned some insider secrets from the experts.
Yeah.
This whole process is pretty mind-blowing.
You know, when you think about it,
Yeah.
cryopreservation is more than just a technique. It's a gateway to incredible scientific advancements.
Right.
It's allowing researchers to study immune responses in unprecedented detail,
Okay.
develop cutting-edge therapies, and even explore potential treatments for diseases that have stumped us for centuries.
It's amazing to think that something as simple as freezing can have such a profound impact on scientific discovery.
Absolutely.
I have one final question for you,
Yeah.
and one for our listeners to ponder as well.
Okay.
Now that we know how to freeze these tiny cells,
Yeah.
what breakthroughs do you think this could lead to in the future?
The possibilities are truly endless.
Wow.
Imagine personalized medicine tailored to an individual's unique immune system,
Okay.
more effective immunotherapies that harness the power of our own bodies to fight disease,
Right.
and perhaps even the ability to conquer diseases that we haven't even encountered yet.
It's exciting to think about the potential that lies ahead. And remember, next time you reach for that bag of frozen fruit or pre-made frozen meal, take a moment to appreciate the magic of cryopreservation. Those tiny frozen particles represent a world of scientific possibilities just waiting to be unlocked. Thanks for joining us on this incredible deep dive into the fascinating world of cryopreservation.